Good evening, everyone. I'm Devika Dhopadkar and I'll be the moderator for today's session. I would like to welcome you all to this webinar on the topic Promises and Pitfalls of Computational Approach in Drug Design and Discovery. Our guest speaker today is Dr. Prashant Kharkar. We would like to extend a warm welcome to you, sir. And before we begin, I would like to give a short introduction about our speaker. Dr. Prashant Kharkar has more than 42 publications, 13 honors and awards, and has worked on 7 projects of various kinds. He also has 2 patents and is also a part of the Royal Society of Chemistry. He is currently a professor at Institute of Chemical Technology and was previously a professor at SVKM's NMIMS. He is a drug discovery professional with an expertise in medicinal chemistry and computer-aided molecular design with more than 8 years of research experience, industrial and postdoctoral. He has managed discovery projects like metabolic disorders, cancer, pain, inflammation in the lead identification and optimization stages. He is currently also involved with things like molecular modeling and synthetic organic chemistry. And now I would like to ask Dr. Prashant Kharkar to begin the session.
Thank you so much, Devika, for the introduction. Let me share the screen. I think you need to, it says who's disabled participant screen sharing. So can you give those rights to me to share the screen?
Hello, Devika. You can make Sir as co-host.
Okay.
Okay, perfect. So is the screen visible now? The PowerPoint presentation?
Yes, sir. It's visible.
Okay, perfect. So then I'll just begin. Okay, thank you so much, IPA branch, and then somebody already started doing things. Okay, perfect. Okay, so thank you so much for inviting me for this webinar, where I'll be sharing my thoughts on promises and pitfalls of computational approaches to drug design and discovery. Now, you might be hearing a lot about drug design and its applications in drug discovery, as well as other aspects of development. There are a lot of myths and there is a lot of hype about these techniques. Very few people are fully aware of its capabilities and the promises, capabilities, limitations, and the promises which these technologies or these approaches deliver. So that's exactly what I'm going to do. I'm going to share the like few thoughts on the capabilities and the limitations of these techniques, and ultimately we probably will arrive at a conclusion where we agree to disagree or disagree to agree, whatever you take it as, that these techniques are mere tools, and their optimum use depends on the human mind, right? So that's what we are going to talk about. So like I already thank the organizers of this guest lecture or webinar, and of course the participants for taking time out to hear what I'm going to share, my thoughts, my experience. So thank you so much for your time. And before I begin, I usually display this slide. The views, ideas, and opinions which are expressed in this webinar, they are solely of mine. They may slightly deviate from what is generally accepted, and that deviation is attributed to my own experience in this field for quite some time, since my ICT days, my master's and PhD days back in 2000, early 2000. I have been a big proponent of these techniques, and I have used use these techniques in my projects, and wherever industrial projects where I worked on, or industry where I worked in, like Piramal Life Sciences, Reliance Life Sciences, and others. So day to day, in our day to day activities, we were instrumental in using these techniques for the drug discovery projects. And then when it comes to drug discovery projects, every project is unique, every project is different, and the kind of problems which you face in every project are of distinct type. Although you can have some generalization, or you can use the learning from one project to the other, but then at times it's not so very straightforward, and blame it to the complexities of the biological systems, and so are the drugs acting on these biological systems for bringing about a particular therapeutic response. And in the process, these drugs will also kind of bring with them a lot of side effects because of their interaction with off targets and a lot of issues actually. So whenever a drug is developed, the purpose is always to show that the drug is more beneficial than the kind of adverse effects or the limitations or the problems with which it has. And on an average, every single drug shows 70 different side effects. So whenever you are writing in medicinal chemistry answers, like a medicinal chemistry question paper answers, so that time you always write nausea, vomiting, dizziness, then lightheadedness, headache, whatever. So most of these occur with almost all the drugs, right? There are very few drugs which are with very less number of side effects. But of course, this is how the way the drugs are discovered. I'm talking about small molecule drugs, and macromolecular drugs, they have their own issues or problems, right? But we'll not talk about it. So today we are going to talk about a lot about drug design and discovery. In the process, I will also talk about some of the terms which are used regularly, sometimes interchangeably, sometimes these are misnomers, as in what people say and what they want to refer to is where these are like two different things. So my first set of questions to all the participants is, are you familiar with drug discovery and development, right? So I'm not talking about design per se right away. So the very first thing is drug discovery and development. If you look at the way drug discovery and development has evolved right from the time when sulfonamides were discovered back in 1935, right from the time when isoniazid was discovered back in 1952, rifampicin was discovered in early 1960s, and there are many other drugs. And then there was in early 1960s we had thalidomide disaster, and then the whole scenario of drug discovery and development with reference to regulators or regulatory agencies, the whole thing changed. So that's where and then the modern day drug discovery is way too different than what we used to do. And then what all things we do in modern day drug discovery, we are going to just briefly touch upon. My next question, of course, related to the first question, that is drug discovery and development. So the next thing is which is part of drug discovery, is drug design, right? So people talk about drug design very commonly, and this is again a fancy term, I must say. People use it for all the wrong reasons, and then with the same notion that whenever you talk about a design, fashion designing, or kind of a building design, or some anything, any design. So that's where people have their own perceptions about drug design. So we'll briefly touch upon that topic as well. Computer aided molecular design. So again, drug design versus computer aided molecular design, which is actually a subset of that. Like CAMD is a subset of drug design, as in drug design has got lots and lots of approaches, out of which one of the prominent or widely used approach is computer aided molecular design. So I'm not saying drug here right away. Earlier people used to call it as computer aided drug design. You don't design drugs, actually you design molecules, and whether they become drugs or not, only god knows, right? And even those who are into development, even they don't know whether the molecule which they have designed, synthesized, tested, and did everything, whether it will become a drug or not, nobody knows. Okay, of course there is some level of confidence in the kind of molecules which are moving from phase 1 to phase 2 to phase 3, but then you never know, you get surprises all the time during drug development. So that's what we will also see. Then again, the broad umbrellas under which many of these design technologies or drug design or computer aided molecular design technologies can be classified or can be put in. So these broad umbrella categories are cheminformatics and bioinformatics, right? Again, you probably know about bioinformatics, or maybe to some extent cheminformatics or chemoinformatics. People use various forms of these terms. So we will briefly touch upon these topics and then eventually plunge into the bigger area, that is so-called drug design or computer-aided molecular design. And then we'll talk about how these technologies have been so very helpful all these years, probably last 5 or 6 decades, and moving forward where where we are and where we would be, say, few years down the line or few decades down the line, and how the nature of these technologies have changed over the years, and what are the issues also, like currently currently the present issues which we are facing with these technologies, and how to, like, what is the turnaround or what is the detour around these technologies, right? So now we are on slide number 4. Slide number 26 is a thank you slide, and in between we have another 22 slides, but then that is enough for me to talk about like next 4 hours, but then we are definitely not going to do that. So probably next 35 to 40 minutes we'll wind up, but then I would want you to understand the terms and eventually take some message out of it, as in keeping in mind that there would be undergraduate students, there would be PG students, probably some faculties as well. So I have tried to keep the tone of the presentation to very low in terms of the technicality, although I can talk a lot without even referring to any of the slides, but then wherever appropriate, I will make a mention of what is the current state of the art and how these technologies are helpful and what are the present issues or limitations, right? So because that's what our theme is, right? So coming back to drug discovery and development, just one slide. You probably hear anytime you hear any speaker who is likely to talk about drug discovery and development, or in general about drug discovery, you will see those fancy slides where at the beginning you have got some target, and then there is high throughput screening, and then you have got combinatorial chemistry, then preclinical studies, and then phase 1, 2, 3 clinical trials. I'm not going to do anything like that. Right away I will give you some numbers which will tell or which will convey the intricate difficulties associated with drug discovery and development. Now, these are two different terms, drug discovery and development. Till the time the molecule enters in preclinical studies or preclinical trials, or I would say in animal studies. So till the time the initial molecules or hits are discovered, till the time they enter in the animal studies. So that part is drug discovery. Then you have got preclinical studies, that is completely in animals. And once all the safety pharmacology as well as toxicity studies in animals are done, and once we are sure that the compound is safe to be used in humans, then we file investigational new drug application, that is a permission to conduct phase 1 clinical trials to the FDA, right? So the local FDA, or could be US FDA or EMEA or other regulatory agencies. So once they review the preclinical data and of course the in vitro data from the initial drug discovery phases, they will make sure that the drug is safe to be used in humans, and then the permission to conduct phase 1 trials is given. So the moment you start with phase 1 trials to phase 2, 3, and after phase 3 trials, you submit new drug application, that is NDA. Now, ANDA, abbreviated new drug application, is different than NDA, so don't get confused there. So once the NDA is submitted, new drug application is submitted, which is the summary of all phase 1, 2, 3 trials, and the data is reviewed by the FDA very religiously. And at the end of it, they come out with a conclusion that in most cases the drug is safe to be used in humans, and that's where you get marketing authorization or the permission to launch the drug in the market. And once the drug comes to the market, to the clinic, again phase 4 clinical trials, they continue till the time the drug is in the market or in the clinic. So to this whole process of bringing a new medicine to the market, according to the latest figures, it takes around 4 billion US dollars. Now that's a huge money actually. So 4 billion US dollars, probably, I don't know, it's this could be anywhere between few thousand crores or something, more than 7 into 4, 28, maybe 28,000 crores or something, I don't know. So according to the whatever is US dollar rate. So you can imagine, such a huge amount of money goes in drug discovery and development. So out of this whole money, approximately 60% expenses are incurred on the development, that is phase 1 to 3 clinical trials, because depending on the drug indication or the use, depending on the severity of the disease, or whether a large population is affected by that disease or disease state for which the drug is being developed. So you can have various number of patients. In phase 1 is the lowest, then phase 2 is little bigger, maybe somewhere around 300 to 600 to 1000. And in phase 3 you probably have few thousand, like 2000 to 10,000 patients, again depending on the disease or disease state. So that's where you need to pay to the patients, you need to pay to the hospitals, you need to pay to the doctors or investigators. So a lot of money goes in that, and that's where whatever money is spent on bringing a new medicine to the market, approximately 60% expenses they incurred on development. Drug discovery, the initial part of it, and the preclinical studies, they take around 20% each, roughly, right? Leave aside the regulatory expenses for preparing the applications and submitting those IND as well as NDA applications. But then this is roughly what is needed for different stages of drug discovery. So then obviously, so if someone is spending 4 billion US dollars, and in fact some drugs they take like 10 billion, $20 billion. Sometimes the drug chemistry is so complex. Chemistry, as in the whole whatever studies they carry out with drugs, they're so complicated. You're not getting good results in one trial, so you go on moving from one trial to the other trial, and the expenses go on piling, right? So obviously people would want a higher return on investment, but then to a great surprise, this return on investment is awfully low. Probably only 5 of 100 drugs which are launched in the market, they bring about whatever money, or they recover all the cost which has incurred in developing them. Others, they just go off the track, and the money is down the drain. That's why if you look at big pharma companies, so they will always have various programs in cancer, in inflammation, and whatnot, and they will have a pipeline of agents. So meaning some agents in phase 1b, some in phase 3, and then strategically they will place this pipeline in such a way that there would be continuous recovery of expenses, or the money keeps flowing, so that they can reinvest that money in for the development of other candidates, right? But despite of this, still pharma industry worldwide still makes $300 billion in profits, right, annually. And most of this money is spent on advertisements, right? Although we don't realize that, or probably we don't do to that great extent in India, but outside, there is there are advertisement made for the consumers. In India, that's not allowed. Consumers, as into the for the patients, right? So the way the pharma market and the whole insurance thing works in developed nations is very different. And that's why US being the first or the topmost or biggest pharma market, so that's where everything is compared with US, and then US FDA is the gold standard of whatever rules they make and whatever regulations they bring into practice. So in all, drug discovery and development is awfully complex, awfully complicated. It's a costly affair, and everybody wants their drug to become the blockbuster drug, as in a drug which will bring, say, at least $1 billion a year. You know but then there are very few drugs which do that, and that's where the dream is shattered for many big companies. And that's where now people have started developing drugs together, as in 2 3 companies, they come together and develop one drug, so that the risk is shared. So even if the drug fails, the risk, whatever money which is gone in the development, is shared. And even if they make profit, the profits are also shared. So a lot of things are changing, or rather have changed in last decade or so, where the whole scenario or the whole model of drug discovery has undergone a big metamorphosis. Not that it has changed completely upside down, absolutely not, but then people are certainly looking forward to different changes. And then there are various reasons for the expenses being shot up, whereas the productivity going down. But then this is how it is. And like I said, only God can tell you whether a person or whether a drug becomes the reality or not, as in the development is probably the most difficult part of it. So that's where people try to make sure right from the drug discovery state that the drug has all the good attributes, and all the issues, whatever issues we are likely, like the drug is likely to have, a drug candidate is likely to have, they are taken care right from the beginning, like right in the drug discovery, so that going forward in the clinical trials, the drug should not fail for whatsoever stupid or some hi-fi reason. And that's where these technologies like computer aided molecular design or related technologies, they have become very important for the same reason, that because if the drug fails early, say in drug discovery, that it doesn't, it cannot further be modified for having all the good attributes, then you just dump that drug right away in the drug discovery stage. You don't take it to preclinical and then early clinical trials, because if you fail in phase 3 clinical trials, so at least eight years have gone down the drain, at least $2 3 billion have gone down the drain or on its development. So that's why they say you fail early, you fail cheap. If you fail late, you fail extremely, that becomes an extremely expensive affair, right? So the next part, drug design. Like again, although this term is very popular, drug design, but then again, I don't personally use this term. I always prefer to use molecular design, be it a computer-aided molecular design or simply molecular design. So molecular design is the proper term, because you can't design drugs. You can design molecules, but whether they become drugs or not, that's not in your hand. That's not in anybody's hands for that matter. But let us talk about drug design, which is a kind of a misnomer, which is very widely used. So basically, this is an approach to tailor-made drugs to suit a particular purpose. Meaning, say now COVID-19, people have been struggling to discover drugs for COVID-19, and given the amount of time which we have, say like last 6 months or maybe more than that, 10 months when it was first discovered, and then the kind of effort which goes in the kind of effort which goes in discovering drugs, or rather this whole process. So, and of course we don't know much about the virus, although we know a lot today compared to what we knew in January or so, or maybe before that. So the more you know about what you want to discover or design molecule for, the better it is to handpick those molecules which are likely to do the job. And that's where, because we did not have much information about the virus, we did not have much time. That's where people resorted to another approach where the approach called drug repurposing. So I don't know if you have ever visited Raymond's work, Raymond's suit or shop, where they stitch the suits. So they are they will they'll probably not take your measurements, they'll get you something to wear, some coat to wear, and they will tailor make that suit, as in, by changing some measurements so that it fits you properly. So that's exactly what a drug repurposing is. You already have a drug, and you try and figure out whether that particular drug is likely to be useful for another indication, say in this case COVID-19 or other SARS-CoV-2, anti-SARS-CoV-2 activity. And then there are many drugs which you eventually must have read about. Many drugs have shown activity. Hydroxychloroquine is a very prominent example. Although there was a controversy, there is still a controversy whether it is useful, but then it's a different thing. So because we did not have much time for COVID-19 for developing drugs, so we resorted to discovery of, or rather fishing out drugs using an approach called drug repurposing, meaning you repurpose the drug, already known drugs. But then let us not talk about drug repurposing here, because the whole point is very different. Okay, so the more I know about for the targets or the organism against which I want to develop a drug, or the disease state where a particular protein is kind of dysregulated, and that's where you need to control that abnormality or dysregulation of that protein, and you go ahead and kind of make a drug which will take care of that pathological process, right? Either inhibit, augment, whatever it is, depending on the context. So you are trying to make a drug which will give you a particular therapeutic effect. And the more you know about the target against which you would want to develop a drug, say in case of SARS-CoV-2, there are two protease enzymes, mpro and plpro, which are crucial for the initial processing of the, after the virus enters inside the cell, epithelial cell. So if you develop inhibitors of those proteases, you are likely to cut short the virus's further journey, getting into the replication stage and eventually disseminating the disease. So that's where the target, which has an important implication in terms of the disease process. So you target that target, meaning you target that protein or whatever macromolecule which is involved. So you develop a particular moiety, a small molecule for that matter, which will go and sit there, and it will disrupt that pathological process. So that's where the tailor-made comes in picture. So this is where the drug design helps you in doing so, right? And again, it depends as to what information you have about the disease process and the proteins or the targets which are involved in the disease process against which you actually are likely to develop drugs. And that's where the drug design comes in picture. So what are you trying to do when you are designing drugs? So everybody would want to talk about potency, meaning you would want a drug to be active to disrupt the pathological process at a very small concentration. And that's what the smaller the concentration, more potent the drug is. So it has a direct implication on the drug dose. Say diazepam, probably zero point 0.5 milligram or maybe 1 milligram is enough for you to put to sleep, whereas paracetamol 500 milligram will be required for bringing about the elevated body temperature. So that's where you see diazepam 1 milligram tablet or zero point 0.5 milligram tablet, and paracetamol 500 milligram tablet. So almost like a thousand-fold difference in terms of their weights, right? So that's where you say that paracetamol is a very potent, the diazepam is a very potent drug. Although I'm not talking about the end effect, I'm talking about the amount of drug which is required for bringing about a particular therapeutic effect. So everybody would want a potent drug, very potent drug, a nanomolar inhibitor, or probably a picomolar inhibitor. So the more the potent drug, lesser is the dose which will be required for eventual therapeutic benefit, because it has direct implication on the cost. Suppose if you want to make a zero point 0.5 milligram diazepam tablet versus 500 milligram diazepam tablet. So you can imagine the cost difference probably would be maybe 100-fold, if not 1,000-fold. So you can imagine potency has got a great implication on everything, because again, if the drug is more potent, that means you will have to give only a small amount of that drug for bringing about the therapeutic response. And then because the drug is given in small amount, the side effects will be also low, right? So that's where everything becomes very apparent. So you want potent drugs. So So you also want to kind of make the drugs more selective so that they don't interact with related targets, or for that matter off targets, so that the side effects are reduced. Because ultimately in clinical trials you're trying to show that the side effects are lesser, or the beneficial effects are more than the side effects, right? So it's always this you have to prove, because every drug will have side effects, but then unless you show that the drug has more beneficial effects than the side effects, you will not get the marketing authorization. So selectivity is another thing. Pharmacokinetic profile, again, if the drug undergoes very high first pass metabolism, then you give 500 milligram of that drug, and hardly anything will be available, right? So that's where you also have to have better pharmacokinetic profiles, drugs which will not undergo, say, very high first pass metabolism, which will be available where they are supposed to act, right? And then of course toxicity issues. There should not be any very typical toxicity issues, because otherwise if the toxicity issue becomes apparent when the drug goes to the clinic after, say, phase 3 trials, at any point of time the drug may be withdrawn. And there are many drug withdrawal examples. Selective cox2 inhibitors like rofecoxib is a very prominent example, right? But then there's a different story about that. Let us not talk about that. So then, who designs these drugs? Like I told you in the beginning, although I did conclude right away that whatever techniques we are going to talk about, they are just tools, right? So ultimately, it is a human mind with the help of computers, or both the human mind and the computer combined efforts will help in designing the molecules which are likely to become drugs, meaning which are likely to have all the good attributes like potency, selectivity, better pharmacokinetic profile, lesser toxicity issues, right? So it's all a human mind, it's the human brain which makes it possible everything. And computer, at least in next till next few decades, will not be able to supersede the human mind, at least in this aspect. Although people try to claim, you'll see probably some news where artificial intelligence, machine learning, deep learning techniques have come up with a drug. But then these are just news line or news heading. I don't believe in that, at least not at this point of time. Maybe 50 years down the line, it may be possible, but then till then, it's the human mind. And then why to design drugs? Of course, to optimize drug properties quickly, rationally, intelligently. And there are so many ifs and buts, or rather I would say decision gates you will encounter during drug discovery. So you will be asked a question whether to go ahead or not to go ahead. And to answer, to take an informed decision at those decision gates, you will need to take help of drug design. And moreover, this drug design technologies or molecular design technologies will help you save tremendous amount of time, tremendous amount of efforts, cost, and everything. And it will try and give answers. It will give you a selection of say 100 molecules from possible 10,000 molecules, which if you end up making 10,000 molecules, it will take like few years, probably a year or so. And then at the end of it, you will still arrive at those top 10 molecules. But here, the drug design technology or molecular design technologies will suggest 100 molecules. And again, you will still be able to get those top 10 molecules from that set of 100 molecules. So you have saved the effort of making those futile 900 molecules, which anyways would not have made it to where they should be, like the preclinical studies or something like that. So these technologies have tremendous potential to reduce the efforts which otherwise would be wasted in doing things. So these technologies help optimize the drug discovery process, or rationalize or systematize the drug discovery process at a very cool pace, right? So the very specific term computer-aided molecular design. So obviously, as the name indicates, it's the use of computers to test hypotheses mostly generated by bright minds. So like I told you, when I say these bright minds could be a chemist, could be a medicinal chemist, could be a phytochemical scientist, as in a pharmacognosy person who is likely to be involved in extraction of like preparation of extracts of phytochemicals, and anybody actually, not just even a biologist for that matter, who observes some very interesting phenomena while studying something during some biological study, and eventually he or she comes up with an idea, and you use the computer, you take help of computers to test the hypothesis whether that compound is likely to bind to a particular target in the binding site, or that is likely to be very similar to other active molecule in terms of the chemical properties or something like that. So that's where the use of computers is very important, and that's where it's computer aided. It's right. So that's important. So here, you are trying to virtually screen the molecules, hypothetical molecules, meaning before even you make molecules in the lab, you essentially can generate hundreds, thousands, or whatever is the number, few millions, billions of molecules, with the help of a small command or a small program, and it will generate those millions and billions of molecules, which are hypothetical molecules. Nobody probably has made them. But then how do I know that out of those 1 billion molecule, which top 100 molecules do I make, so that my probability of identifying a molecule which is likely to be very potent and selective for the target at hand increases to a great extent. And that's where these hypothetical molecules can be virtually screened, or screened by a computer, or screened inside a computer for a particular end effect, for binding to a target or for anything which you want to study, right? So that's where computers help, and with the help of the computational power which is available today, and the kind of advances we have in various basic sciences including physical chemistry, it is now possible to do a lot of things with great surety, and that's where the promise of these technologies lie, right? So here you're trying to screen these hypothetical molecules for potential similarity with experimentally screened active molecules, meaning suppose you isolate a phytochemical which is likely to be very, very potent anti-cancer agent, but then because it's a phytochemical, it's a very complex natural product molecule which has got, say, 10 different chiral centers. So the possible stereoisomers would be around 2 raised to 10. So probably 1,000 molecules or so, 1,000 stereoisomers, which are extremely difficult to make it in the lab. So in that case, what I can do is I can try and look for molecules which are simpler, but then which have got a similarity to the active anti-cancer molecule, right, which has been experimentally screened. So here I'm not talking anything about the macromolecular target, because in the earlier approach, which is called as structure-based drug design, where the three-dimensional structure of the target is available, but we are encountered with many, many projects where there is no experimentally determined structure of the macromolecular target, or we don't even know what the target is. So forget about doing the structure based drug design. So in that case, I would be interested in a simpler molecule which is very similar to the molecule which is active in the experimental settings, right? So that's exactly what I will do, and that approach is called as ligand-based design, and that's what we are going to do. So you probably might have seen pictures, very fancy pictures like this, and this actually represents the so-called structure-based drug design. So all those fancy ribbons or those purple loops or tubes, those actually represent the protein part, and in between in those stick format, some molecule. Now I think this is very similar to celecoxib or something. But anyway, this is not a hypothetical molecule. This is a true molecule, and this is actually this is from X-ray or three-dimensional structure of the small molecule, that is the inhibitor of the target, which is represented by the protein in the active site, right? So those dashed lines basically show the interaction between the protein and the small molecule. There is this red ball that is actually a water molecule which is interacting with the molecule and also interacting with the protein, so water mediated hydrogen bonding. So basically there is a cavity inside a big protein, a small molecule which has got a great shape and electrostatic complementarity. It just goes and sits there, and then you picture that particular pose where the molecule is bound to the active site of the protein. And this is what they do in structure based drug design. So basically they will crystallize the protein, they'll produce the protein in by rDNA technology, they'll crystallize it, and then they'll soak the crystal with the small molecule inhibitor which is known from the experimental assays. and eventually you picture, like you solve the crystal structure, subject it to X-ray diffraction, you get the diffraction pattern, and using very high end techniques you solve the structure and see these pictures. So this actually tells you that there is a pyridine ring in the molecule which is involved in hydrogen bonding with a water molecule. So if I convert that pyridine ring to benzene ring, meaning that pyridine nitrogen is gone, then in that case the hydrogen bond with the water molecule is gone. So the potency or the affinity with which the molecule will bind to the target will be reduced. So basically you get a lot of clues. So now that you have a mold of the active site of the protein, so you can go on putting virtually with the help of some sophisticated softwares or programs, in a process called molecular docking. You go on putting the molecules of your choice and see whether they're able to mimic those interaction or bind to the proteins with high affinity, and that's where structure based drug design is. There's another approach which is called as ligand-based drug design, and pharmacophore elucidation is one such technique. So where you have got active molecules, you align them, and then you extract this. Tell the software to extract the common features of which are common to all the active molecules, and these pairs, what you see named as HBA, HRA, and all those. So that basically tells you that these are the features which are required for activity, and those features should be located from each other at a particular distance, and those distances which you see here, 8.35, 5.11, 6.22. So basically, this figure tells you about the geometrical description of the features required for activity. So if I have that information, I can probably tell computer to design molecule, or probably generate a data set, a hypothetical data set of molecules, and then the computer will go and matching the molecules with these set, and wherever there is a match, it will give it as a hit, and that's exactly what is shown here in the figure B, where a molecule which has all those features located at a particular distances. So this molecule which has got a pharmacophoric similarity with the pharmacophore is likely to be active. So this is a ligand based drug design where we do not take into consideration the three-dimensional structure of the protein at all, because we either don't know or we don't want to use. So sometimes these both approaches are combined. So even if you know the three-dimensional structure of the protein, you still go ahead and use ligand-based drug design and combine both, so that the robustness of the outcome is increased, right? So the next term is cheminformatics. As the name indicates, it's a discipline which deals with chemical information. So the moment you say chemical information, say when you talk about drugs, a small molecule drugs, so everything is dictated by the structure of the drug. So how to represent this structure in the form of some numbers, because I have to use this chemical information which is kind of present in the form of a structure, so that I can use that information for variety of data mining techniques, and that's exactly what is done in the cheminformatics. So you represent the chemical structures in the form of some numbers or descriptors, and then use these numbers for developing models and predicting the important properties by of the molecules, maybe hypothetical molecules, by computers. So in a way, cheminformatics in raw terms is called chemical data mining. So you have structure, you have probably you also have activity data, and use this cheminformatics or chemical data mining techniques, and make some meaningful extraction of information, and use that information for prediction of molecules which you are likely to make, some hypothetical molecules, and with the help of cheminformatic analysis, you will get your answer just like you otherwise get answers with the help of say molecular docking. Out of 1,000 molecules, you make only 100 molecules, and out of which 10 are active. So just like that, right? Now, as far as cheminformatics is concerned, in last 5 years or so, or probably a decade, a little less than a decade, machine and deep learning approaches have become very, very commonplace in a variety of cheminformatics analysis, be it toxicity models, be it pharmacokinetic models, be it some activity models. And then there are many, many free softwares available or web tools available, which at the back end of it is some machine learning or deep learning model. All you have to do is just put the structure or the smile string of the structure, and it will predict what is the what is likely to be the fate of the molecule, whether the molecule is going to be toxic, non-toxic, pharmacokinetically more like better than maybe the other molecules, meaning human intestinal absorption or PGP substrate. Any and every property you can determine. Even the activity can be determined. So that is cheminformatics. Similarly, bioinformatics, which is another important term, it's a discipline which deals with biological information. Just like we had chemical information, here it is biological information. The moment you say biological information, so it specifically deals with the genomics or the information which we get using analysis of genes. So if you have got a particular disease or disease state, so what are the genes which get altered in that disease state, say something like cancer disease state or disease, and you compare it with the healthy individuals, and then you derive the genetic signature or genetic basis of the disease, and you focus on those genes which are kind of dysregulated for discovering drugs or for developing drugs, right? So that's, in a way, it's a biological data mining. And now, given the amount of genetic information available, you can imagine the kind of data which is eventually generated from all of these techniques. So again, here the machine and deep learning approaches, they have become a very commonplace in bioinformatics as well, right? So wherever there is a huge data, wherever there is a huge data, these machine and deep learning approaches and artificial intelligence based approaches are likely to make their mark or leave their mark in the data analysis, right? Because these are data mining techniques, right? And that's where people are kind of using them at a very on a very large scale. Of course, thanks to the availability of high-end computers or supercomputers, because if you're dealing with, say, 10 raised to 12 molecules, then you can't really do it with a computer which I'm currently sitting in front of, a laptop or so. You need workstations. You probably need supercomputers to deal with those kind of heavy duty computations, right? So now coming to the earlier version of CAMD. So basically, back in 1960s, particularly in 1964, quantitative structure activity relationship was introduced by Professor Corwin Hanch and then Fujita Ban, right? So that was the era when it all started. Probably before that, it was all random screening, meaning suppose if you want to develop an antibacterial drug or antitubercular drug, you go on making whatever you can, whatever is possible in the lab. You go on making those compounds, random screen them. If you get some molecule out of that, and then you further optimize that molecule using structure activity relationship. But then with the help of QSAR, people started focusing on a particular chemical series and the trends which essentially are responsible, trends in the structure or the variation in the structures, which essentially can help you get an optimum, a drug with an optimum set of properties, including pharmacokinetic properties, toxicity properties, activity properties, like that, right? So then in early 1980s, structure-based approaches started appearing in the literature. So they started protein data bank, and then initially there were only few molecules, few macromolecular structures. Bacteria rhodopsin was probably one of the first macromolecules whose three-dimensional structure was solved by X-ray, and then there were some structures by NMR solution structures. And then after 1984, when rDNA technology was invented, so it was possible to make proteins in larger quantities, and because of which the crystals, protein crystals, were developed because the protein was available, pure protein was available in larger quantities. So eventually they started making more and more protein crystals, and subject to X-ray crystallography. So slowly the number of structures in the protein data bank or PDB went on increasing, and a few years ago it crossed 100,000 mark, and it is very close to 200,000 structures. Now the speed with which it is being done is tremendous, right? If you look at the number of structures deposited in PDB over a period of, say, like before 2000 and after 2010, there is a sharp contrast in these two things, right? Like two era. Of course, because of the advances in X-ray crystallography called high throughput X-ray crystallography, and of course the computational power. And currently, We also have another technique at our disposal, which is called as cryo-electron microscopy. In 2015, three scientists were awarded Nobel Prize in chemistry for their advance or for their advent of cryo-electron microscopy. And in fact, I think two or 3 weeks ago, there was a news in some magazine that nowadays, with the help of cryo-electron microscopy, it is possible to get atomistic details of molecules. Now, that's a huge accomplishment. So, read more about cryo-electron microscopy, and you will be amazed the potential which it has. You have studied the TCA cycle or Krebs cycle. Probably this year or maybe earlier, and all those fancy complexes like pyruvate dehydrogenase complex and all, so their structures were never known because they are like huge proteins, a conglomerate of 5 or 7 proteins. But now we know their structure, their three-dimensional structures, and thanks to electron microscopy, and of course, the kind of chemistry knowledge or the chemistry expertise goes behind that, including the computational power which supports visualization of those structures. So all of these advances in various areas of science, they have spurred the growth in these very important areas of life sciences, which has made possible to discover many things. And another important thing was completion of Human Genome Project back in 2003, when I just graduated, like my PhD was just completed. And after completion of Human Genome Project, the number of targets which were available for therapeutic intervention, they went from less than 500 to more than 5,000 or so. So you can imagine, suddenly we had this information available that a particular target could potentially be used for developing drugs for intervening a particular disease process. So that was like, it's like, you are walking or you are in a bullock cart, and suddenly that bullock cart turns into an Audi, and then you are asked to ride at a very high speed, because like in 0 to 100 miles in 6 seconds or less than that, 3.2 seconds or so. So you can imagine the speed with which we were going, the bullock cart versus an Audi. So the speed with which the science was kind of forced to progress was phenomenal, and we are still trying to cope up with the speed with which these things can take shape, and make us aware of many fundamental aspects of biology, which ultimately everything goes to drug discovery, right? Be it human drug discovery or be it veterinary drug discovery. Although veterinary drug discovery is always kind of on the other side, because many of the drugs which are discovered for humans are used in veterinary practice or for animals, but then they also have their own drug discovery centers, right? But then not many companies do that. Okay, so all these years, this whole technology has matured, although it is still evolving, but then it has reached a bit maturation level, because now we are able to do a lot of things, like the predictions are more accurate compared to what we used to do 2 decades ago, right? So people use multiple hybrid approaches, structure-based, ligand-based, machine learning, and whatever, whichever gives you better predictions and better reliance as to how the results or outcome will turn out to, and that's where people end up using. Availability of sophisticated softwares, which was not possible before 2000, now it is possible, and earlier which were just a dream of a medicinal chemist or a computational chemist, now those are realities. And of course, cryo-electron microscopy I already mentioned about. So the availability of these structures have spurred the growth from bullock cart to an Audi, and that's where the whole thing is revolving, and trying to come up with greater success stories. So coming back to CAMD success stories, right in 1984, when AIDS was first discussed, or rather discovered, that it's a deadly disease in humans, just like we have COVID now. Of course, it wasn't that bad for AIDS. I was probably just 6 or 7 years old when this thing happened. So during that time, people started taking help of computer-aided molecular design for HIV-1 protease inhibitors or reverse transcriptase inhibitors. And usually, discovering a drug takes around 10 to 12 years. But these initial HIV protease inhibitors as well as reverse transcriptase inhibitors, they were discovered in a record time of 4 to 5 years, right? Of course, because we needed the drugs badly, to curb to control the spread of HIV, because then it ultimately had other complications like TB and other diseases because of the lowered immunity. So eventually, they took help of HIV protease structures, HIV reverse transcriptase structures, and the associated computer-aided molecular design approaches which were available back then. And then they developed even the crystal structures which were available for HIV protease, reverse transcriptase. So they came up with the drugs in record time, and that was probably one of the earliest success stories. Although rational drug design success story, the first story they talk about is discovery of nalidixic acid and the DNA gyrase inhibitors like ofloxacin, pfloxacin, ciprofloxacin. So those in the early 1980s, those are actually regarded as the rational first set of rational design drugs, right, floxacins. But then I would still regard HIV-1 protease as the one of the earlier success stories. Then came imatinib in 1995 or so. Imatinib, whose trade name is Glivec, it was discovered by, I think, Novartis, for chronic myelogenous leukemia, a blood cancer, type of blood cancer. And this is actually a first rationally designed drug where they came right from the beginning, where they screened the first set of molecules. So they used the crystal structures of BCR-ABL protein to come up with imatinib, and imatinib is still a blockbuster drug, although you have got other versions of imatinib or other other congeners, the same series molecules. But imatinib is still one of those earlier discovered drugs, although there are many cancers, many blood cancers which are resistant to imatinib. So, but then the point is, if you read about imatinib, how imatinib was discovered, it's very, very interesting, and it is one of those brightest success stories of computer-aided molecular design. Then, apart from structure-based drug design, or even for that matter, ligand-based drug design, the computer-aided molecular design was used for optimizing the pharmacokinetic as well as physicochemical properties. So, in fact, in 1996, Lipinski's rule of phi was published. Before that, the drugs which were used to undergo attrition in late stage clinical trials like phase 2 or phase 3, one of the reasons was poor pharmacokinetic properties. So after that, the whole say after 2000, after Lipinski's paper was published in 1996 or 1997, they actually came up with a variety of I would say approaches, including computational approaches, where they would generate a large number of data points experimentally using high throughput assays, use that data for developing a computational model, and use that computational model for predicting the activity of molecules, activity as in the physicochemical and toxicity properties even before synthesis. So if the activity or the physicochemical or toxicity property is bad, then don't drop that molecule, so that you don't unnecessarily make a molecule which eventually will fail in subsequent trials, including the preclinical studies. So that actually revamped the whole drug design industry discovery scenario, and that actually is still continuing. It's just that instead of those typical supervised learning approaches, now we are using unsupervised learning approaches, including some of the artificial neural network, machine learning, as well as deep learning tools. The tools are changed, but the philosophy is still the same, right? So that's where the ML and DL tools have really come handy, and they are kind of helping in a big way to optimize a large number of properties right in the discovery phase, so that the probability of failure in the late stage clinical trials is very close to zero, right? That's where the CMD and the magic wand. So people always thought, those probably who don't know much about CMD, they always thought that you see some very fancy pictures on a computer screen, which tells you that how a molecule binds in the active site. So that means the molecule, the technology or the tool is such that you just kind of move the wand, and it will give you the drug. So that was the very very misconcept. And right from the time I started working in Piramal back in after my PhD back in 2003, I remember, in fact, a particular incidents. We were working on some, I think that was PPR gamma modulators, peroxisome proliferator receptor activated gamma. So then, the chemist had a molecule which they have synthesized, and I was, I freshly joined back then in 2003. Um, Early 2004, actually March 2004. So I did molecular docking. I showed them that this is kind of binding nicely, and then they got so excited that instead of taking for in vitro screening, they directly took it to the animals and it completely failed. I tried to explain them that this is not how it works. You have to first test it in vitro studies and then modify it so that it goes. So that molecule failed because probably it did not have it did not have good pharmacokinetic properties, although it turned out to be a good molecule in vitro. So that's where the pharmacokinetic properties, they understood the importance of pharmacokinetic properties like cell membrane permeation and all. So there are innumerable success stories. Like if you take Journal of Medicinal Chemistry or any medicinal chemistry or drug discovery journal, you will see so many stories about this. In fact, in recent this week only, there is a success story of CMD published in Journal of Medicinal Chemistry on thrombin inhibitors. Now, thrombin is one of those proteins in blood, right, which has something to do with the blood clotting. And then people have worked for like last 5 decades or so. They never discovered a drug which would act as a thrombin inhibitor, but somebody actually came up with a good thrombin inhibitor with the help of, part of it was based on CMD. And so 5 decades they struggled, and finally CMD could actually give the answer. I thought of including that as a success story, but then I thought maybe you can read more all by yourself, because if I focus on one particular case study, then the whole time is lost in that. But anyways, there are innumerable success stories, and this one is from my own work back in 2012. Before I moved to NMIMS, I used to work for a company, a Dutch company called Nycomed Pharma. So with the help of computer-aided molecular design using structure-based as well as ligand-based design, so we actually listed or selected 50,000 molecules from our in-house set of physical molecules. Around 900,000 molecules. So we zeroed down to 50,000 molecules, and all those molecules were screened at GVK Biosciences in a high throughput assay or moderate throughput assay. And out of those 50,000 molecules, at least 25 molecules turned out to be very good in terms of, and this is what the paper talks about. So here we used all the approaches, almost all the approaches, including pharmacophore, including drug design, including cheminformatics based drug design, as in structure based drug design, pharmaco ligand based drug design, as well as cheminformatics based approaches. You should read this paper if you are interested. So the point is, with the help of a variety of approaches, we were able to come up with a structurally diverse set of molecules. And in fact, this particular target IMPDH2, it's a difficult nut to crack. We actually burnt our fingers in discovering drugs for this or discovering molecules for this. And so I still work on this target, and we have got a lot of different molecules acting on variety of IMPDH molecule, like from human, from mycobacteria, from H. pylori. So that's okay. So another aspect, as far as, so we will now slowly move towards the end. So now my tone will be more pessimistic, as far as well, not exactly pessimistic, but the realism actually will now pip in. The hype versus hope. So all these years people were always kind of very excited about these technologies, but then not that these technologies would work every time. There are basic fundamental differences when it comes to simulating something which is happening in a very complex biological environment onto a computer screen, right? And that's where one has to realize the difference between hope versus hype. So these technologies, by those fancy pictures and kind of success stories, they all created a hype, but then when it comes to the realism, many times these technologies wouldn't work. A molecule which is likely to be predicted like to bind very nicely with the target wouldn't show any activity in vitro assays. So again, so we were basically missing something. And one of those issues was oversimplification of complex biological events on a computer screen, right? And that's an example is ligand receptor bind. So you see in this particular diagram, a biological target, when it is inside a cell or be it on the cell surface, so it is always surrounded by water because everything is immersed in water, everything is water, right? So when a molecule, a ligand binds to or rather approaches to a biological target, the ligand is also surrounded, it's in dissolved state, so it is always surrounded by water molecule. So when these two have to bind together and form a complex which will lead to some effect like inhibition of that target, so first that layer of water molecule must be withdrawn from the biological target as well as ligand. But then this doesn't, in thermodynamics terms, it doesn't happen so easily. It's basically there is a penalty, energetic or entropic penalty associated with that. So if your molecule, the ligand, is binding with the target and forming a complex, so whatever is the complex energy, that has to be much lower than the individual energies of the sum of individual energies of the macromolecule or the target and the ligand. And there is a negative aspect to that, that is the desolvation from target, desolvation from ligand. Because if that is not the case, then it's not a spontaneous process. So in thermodynamics term, it will not really happen, right? Because you're trying to simulate it at 37 degrees, that is at body temperature. So this oversimplification sometimes leads to a false outcome or false prediction, and that's where one has to be very well aware of that. Then another important aspect which many of these computational tools they fail to understand or fail to mimic is the macromolecular conformational change upon ligand binding or during ligand binding. Meaning, when a ligand approaches to the macromolecule and binds to it in the active site or binding cavity, so there will always be some conformational change associated with the macromolecule, and at times it is very difficult to mimic those or to simulate those changes in the computer. And those changes could be very crucial in terms of making a prediction whether a ligand is binding with the macromolecular target with high affinity or not, right? Which we call it as induced fit. And that's where these predictions kind of at times fall apart in terms of accuracy, because what we are using for doing say molecular docking is a static crystal structure, just one state of the protein structure, which may not, which when it is in a solution, it's always in the dynamic state, although the overall structure remains same, but then the smaller changes in the binding cavity where the residues are kind of flanking here and there can have a big impact on the binding of the ligand, which it predicts the crystal structure or docking predicts that it is binding very nicely, but in real life it doesn't happen. And then there's a very you need to probably take help of some biophysical techniques, some experimental tools to understand that, or something like isothermal calorimetry, right, ITC, biophysical techniques. But at times it's very difficult to simulate on computer, and that's where the predictions fall apart, meaning what you predict will not actually happen, and then it will not be experimentally confirmed. Now this one is a very recent structure of capsaicin receptor, TRP V1 or so, or V7. So basically this is the heat sensitive receptor, and so now you see there are four domains. A transmembrane receptor. So capsaicin, which is present in chilies, it binds to these receptors, and that's where you get that hot feeling, right? So at times these receptor structures can be limiting, and we don't understand them fully, and that's where our predictions are not proper, right? So again, when you're trying to simulate binding of a ligand with its molecular target, although you know what is exactly happening, but then the mathematical equation which tries to catch or hold of all the interactions, say hydrogen bonding, hydrophobic interactions, or electrostatic interaction, when a small molecule binds to its target, whatever interactions which are happening. Now those interactions essentially cannot be tapped or cannot be tracked by the scoring function or a mathematical function which tells that how good one molecule is binding with the receptor and how good is the other molecule binding. So basically it generates some number, and those numbers may not actually represent all the components of binding, and that's where people use a variety of scoring functions. And this is just the overall process process of doing that. So there are a variety of scoring functions like force field, empirical, knowledge base, and consensus scoring, which is a combination of all. So to actually generate a scoring function, you need to train. It's like a mathematical model. So you basically take experimental data from from the actual molecules, and then try to see how the molecule, how the scoring function is able to score that interaction. Meaning molecule A has got a potency of 1 nanomolar, molecule B has got a potency of 10 nanomolar, molecule C has got a potency of 100 nanomolar, molecule D has got a potency of 1 micromolar, right? So tenfold, like 1 log unit difference. So whether the scoring function is able to differentiate between each of these categories. So this is exactly what they train the model, the mathematical model, so that it can identify whether, if you have got say 10 interactions present, so whether you will classify it as a molecule which is likely to be 1 nanomolar inhibitor, or rather its IC50 is 1 nanomolar versus 1 micromolar. So you actually use the experimental data to train the mathematical function, and in turn the experimental data which is used for training this mathematical function itself is variable, because it comes from biological systems, and biological systems are complex or complicated, and that's where the whole things is taken for a toss, right? Although we know for sure how to increase the accuracy of scoring functions. One way of doing is that you combine a variety of scoring functions so that you are increasing the robustness of the outcome, and that's where the consensus scoring, which is a combination of probably all of the better scoring functions which are based on different philosophies. So that's where you are trying to do something, trying to develop a model for predicting something, but then that the data on which the model is based, that data itself is variable, right? Because it comes from biological system. So obviously all the errors are introduced in the model, and then when this scoring function is used for predicting the activity or binding affinity of a molecule to a particular protein structure, then all those errors will propagate, and then you may not actually get a proper result. And that's where it's very difficult to get a correlation between the experimental binding affinity and the docking score. I can reward anybody who can show me that there is a correlation between binding affinity, say like PIC50, and the scoring function or the docking score. I will give you whatever you would otherwise ask from me, right? So it's very difficult. So so ultimately, in all this process, what I'm trying to do is I'm trying to train a model based on a data, but that data itself is flawed, meaning with errors, and that's where you need to deal with those errors, and that's where you need to be aware of the limitations of the technology, of the tool, in terms of its prediction power, right? Although these some of the scoring functions will do excellently well for a particular type of protein, say kinases, but they may not do so well for ion channels, or maybe for other transmembrane receptors like GPCRs. So it all sometimes these models could be very biased, and that's where one only has to be aware of the limitations, and then taking these limitations into keeping these limitations in mind, you have to rely on the results in that proportion, right? And that's where things are kind of, I mean, the promises fall off offered by these techniques. But again, the picture is not that rosy, or picture is not that bad at all, meaning we always have, like I told you in the beginning, every project is different, and every project teaches you some or the other thing. So your mind, your human mind component will always play a role in understanding what is given by the computer, and to what extent you should be believing that, right? So this is what happens. Right, so you are trying to develop a model, and like any model, including a business model, where the models are never constant, meaning every third or fourth month the business model changes. So likewise, you also have these models changing with the availability of new data, better structures, right? So that's where you need to actually take help of this whole or rather understand this whole thing in that perspective, with those limitations in mind. Another interesting problem is protein structure prediction problem, meaning even though we have so much advances in physical chemistry, thermodynamics, computational power, but then it is still so very difficult to predict a protein structure with the help of computer. Suppose if you have a protein of 100 amino acids and you want to understand how this protein folds for the given sequence in body that happens in few milliseconds, right? Not even like hundredth of a second or so. But then if you have to simulate this small protein of 100 amino acids, it's folding in computer with the fastest supercomputer in the world today, it will still take around 200 years to simulate how this protein will fold in computer, like in silico. But then that happens in like few milliseconds in body. So you can imagine the gap between us, our understanding, and the nature, right? So that's where the today's computational tools are not really equipped. So this is what is shown here. The mRNA binds to ribosomes, and the protein it goes on adding all the codons. The protein gets made up, and then these molecular chaperones will help in folding it properly. But if it gets misfolded and it goes to other states, but then these molecular chaperones will eventually bring it back to the properly folded protein, because if it doesn't fold properly, it will not be able to show its or execute its function because of the improper 3D shape. So that's why these things happen in few milliseconds with utmost precision. But then to simulate that using a computer, it's very difficult, right? We have some understanding, but we are still far away from predicting the protein structure. So that's where people try and talk about comparative protein modeling or homology modeling. And so this is particularly done for proteins for which the three-dimensional structure is not available. Say something like I would say membrane protein, because the moment you take the protein out of membrane, its structure, three-dimensional structure collapses, and you are not able to kind of do it. But then with the help of cryo-electron microscopy, it is now to a great extent it is possible. But then nonetheless, the comparative protein modeling or homology modeling still is a challenge by today's standards. And in fact, this is from my own work, where we tried to generate a homology model for a membrane protein called system XC, which is responsible for transport. It's an antiporter, so it transports the glutamate out of cell and cysteine inside the cell in opposite direction, and this cysteine gets converted to cysteine, and this cysteine is eventually is responsible for the its effects, protecting the cancer cells from the effects of reactive oxygen species. So basically, if you have inhibitors of system XC, so they are likely to be anti-cancer agents as well as anti-epileptogenesis or anti-epileptic drugs, right? So the structure is not known, and it's a complicated protein. So we try to develop the comparative protein model and use this model for kind of molecular docking or structure-based drug design of the molecules which were synthesized in lab. And to a great extent, we were successful, but still, like I said, we knew the problems with our model, and that's where of course there was some agreement, but most of it was not in agreement with what our hypothesis was. So what I'm trying to tell you is, to some extent we were successful, but to a larger extent we were not, right? So of course we were aware of the issues with the protein model itself, so we were not at all disappointed. So whatever it could help us in terms of developing our molecules, that was great actually. So likewise, ligand-based design, it offers a great promise. So again, this is from my own work, where this is for dopamine. It's a pharmacophore for D3 receptor. So we took all the active molecules, structurally diverse active molecules, aligned them, extracted the pharmacophore, and used these pharmacophoric features for discovering molecules which were binding or which were matching with the pharmacophoric requirements, and which also exhibited the dopamine receptor binding activity, dopamine D3 receptor binding activity. So in a way, ultimately we use this ligand-based tool, that is pharmacophore, for discovering or for screening molecules which were kind of anti-Parkinsonian drugs actually, right? So and then another one is obviously QSR based approach. So basically these are all like you generate, it's a model, it's a mathematical model. So you have got an activity which is a dependent variable, and on the right hand side you have got a collection of independent variables, and there are some statistical parameters associated with that, say like something like correlation coefficient or coefficient of association, and then the F value, statistical significance, cross-validated R square pred. So based on these parameters, you kind of use one model for predicting the activity of molecules before synthesis, right? So likewise, there are but again, because this is a model, and there is always the error associated with the biological measurements onto which this model is based, right? The experimental data. So obviously those errors get percolated in the models, and then although these models do a good job in terms of predicting the activity to a great extent, but then don't always, we are never disappointed if it doesn't work. But then at least 60% of the times, or maybe 70% of the times, the predictions will be accurate, which is good enough. But then although this is pretty old, this one is from my own paper back in 2002, my first paper in Journal of Medicinal Chemistry. But then nowadays the predictions have become very, very robust with the help of say neural networks like deep learning, machine learning techniques. So you see, all these years there has been a continuous improvement in the quality of models. Of course, the data on which these models are based, there also the data is also very nice, relatively error-free data, and that's where this whole. So we are from a very naive situation, or from a naive state, we are getting into near perfect state. We'll never get into that pure perfect state, but then doesn't matter, as long as our predictions work with the help of these fancy approaches, doesn't matter, right? Ultimately, if at all it is giving me results and saving my efforts of making 900 molecules, all these fancy technologies, these are worth. And that's where you must be aware of machine learning, deep learning. They are entering into every single field of life sciences, material sciences, everywhere. So this is the future. And if you, as a student, if you get a chance to learn these techniques, please feel free to learn these techniques, as in be eager to learn these techniques, because you never know, these techniques would be helpful to you at some or the other point in your moving ahead in your career. Unless you want to set up your own kirana shop or a grocery shop or vegetable shop, there also you will still use it, because you're using some kind of payment option or something, whatever. So basically what I'm trying to tell you is these techniques have become an integral part of our life. And then despite all the limitations and pitfalls, they have come up, these techniques have come up with a trustworthy option or discipline, right? And their accuracy has increased over a period of time. Various success stories. So newer methods, better input data, accurate biological measurements, greater computational powers, and then these fancy approaches like ML, DL, artificial intelligence. So everything has contributed to enhanced accuracy, and we are fairly good at predicting some of the biological endpoints like reproductive toxicity or developmental toxicity, although a lot more needs to be done. And with every passing day, these predictions will be perfected, and the day is not far when we probably would be able to replace many of the animal studies with these models. As in, of course, that's again a distant dream. It will not happen just like that. Many years must pass before something like that happens. So ultimately, many of these models will be an alternative to experiments. Not everything though, but at least some of the high-end experiments where toxicity needs to be predicted, or some other properties need to be optimized. So that's where these models and these techniques come handy. So that's all about it. Thank you so much for patient listening. And if you have questions, please feel free to ask. I have already exceeded the time, but then that's what happens always. You're talking and then you just lose it. I'm so sorry. But yeah.
Thank you, sir, for sharing such valuable insights with us. The platform is now open for discussion as we begin with the Q&A session. And if anyone from the audience has any questions, we have enabled the chat section. Please type your questions in the chat box. Okay, we have some questions already. The first question is, can you please suggest some free or open softwares which help in drug designing?
Okay. See, now if you just type, if you are a totally novice in this area, so you can start with something like Mole Inspiration. Now, if you just type mole inspiration, it will take you to its website, and then there you will be able to see, on the left-hand side, second option, calculation of molecular properties. So all you have to do is draw the structure, and it will predict a lot of molecular properties, which are very basic properties. But then once you build an understanding from that, then you can go get into some high-end molecular docking tools like Autodoc. Now, Autodoc is one of the highly regarded tools for molecular docking. So Autodoc is one where you can get into that. As far as property prediction is concerned, another important site is Swiss Admi. You just type Swiss Admi, it will take you to their website, and all you have to do is either paste a smile string, or you can draw a structure, and eventually it will give you all the ADME properties, toxicity properties, including the pharmacokinetic, say drug absorption, PGP substrate, PGP inhibitor, CYP inhibitor, CYP substrate. So all of these will you will be able to understand. There is another option there, target prediction or target fishing. So again, you have to paste the small molecule structure there, or draw the small molecule structure, and it will predict if it's a totally new molecule. It will be able to predict what could be the potential target, macromolecular target for that particular small molecule, which is actually a great, it's actually a big deal, right? So that can help you in designing a particular biological assay for the kind of molecule which you are working with or dealing with, actually. There are many others. I mean, you start, there is another one from, there is a software called TEST. That's for toxicity prediction. It actually gives you at least 10 different toxicity endpoints. All you have to do is draw the structure, everywhere structure, and you can build up on this particular thing. As in, you will read more, and then you get into more and more of these free tools which are available on the internet. Or you can write to me if you have a particular thing. Apart from this, I can suggest a few more, actually.
Okay, we have another question. There is a saying that rules and are barriers to innovation. Is it true or not?
But in what context, if the person can elaborate? Rules as in, you mean rules as in, say, Lipinski's rule of five or something? If you mean that, yeah, to some extent, when it comes to innovation, it should be free. Meaning you should be able to explore it freely. But then, at times, if there are no rules, you probably would wander in the wonderland, wondering as to what to explore. But then these rules will basically give you some direction, which essentially can explore you in a more focused way. So, yeah, I mean, the extent, the statement is kind of true. But then when it comes to innovation, if the innovation is not focused, then you probably would spend your lifetime innovating something which you don't know what to innovate. Meaning, these rules tell you about a boundary, which you probably can go beyond the boundary, provided you are really focused and try and do things which nobody has ever thought of. So yeah, kind of. But interesting question, but difficult to answer.
Okay, we have one more question. Contract research organizations are very fast growing now, and drug discovery programs are exclusively using these services. Is this
Yes.
culture able to produce the drug flow as expected?
Oh yeah, yeah, definitely. In fact, see what happens is, many times, like if you look at California Bay Area, there are various companies, various small pharma companies which only work on probably one target or maybe just one molecule, very focused ones, right? So what happens is, if you are too focused, and then if you're a small company, then at times it's not possible for you to do everything all by yourself, unlike big pharma companies where they've got big units, and then they do things day in and day out. So if you have something very promising, say a promising molecule, could be a natural product, could be anything. So instead of doing it all by yourself, you tell the people who are trained in that area, and they'll do it for you for whatever fees they ask for. And because they are experts in that field, because they have already set up, you don't really have to invest heavily in preparing or getting that setup ready in your own premises. So So you just give them a fee for whatever you ask them for, and because they are experts, everything is ready, they'll be able to do it, do it in a very fast manner. And this is very, very productive actually. And so this is like, people don't try and invest in capital resources, rather they will go hire the premises, do the job, and then bring something to the market, make the profit. So I mean, you're not really spending on capital premises. And these CROs definitely have got the advantage of in terms of time efficiency and cost. So yes, this is a perfect culture, unless you have that kind of money and willingness to do it all by yourself. But then there are CROs for almost everything, and they are fast, so that will only increase the speed with which the innovation can happen. And in fact, many pharma companies, they don't really innovate. They will go to these small companies, like I told you about, very focused companies. They'll buy out those companies like big fish eating the small fish, and then that becomes their innovation. And that's a very widely accepted practice, at least in the West. In India, of course, we are just kind of getting into that mode. But then that's a win-win situation for everyone, where you don't incur very hefty expenses in doing things which otherwise could be done by somebody in in a fast manner, at a probably a low cost. Yes, yeah, that's a great thing. In fact, I'm myself involved in one such project where we have done a lot of things from CROs, including the efficacy studies, including all the preclinical studies. Because the company for which I'm a consultant for, they don't have these kind of resources. They have in vitro biology, but then the preclinical, they can't do it all by themselves, because that's not the focus. The focus is to bring the molecule to the market, and that's exactly what we have done in last 2 years or so. And soon we will be filing an IND application for that.
So the next question is, can you suggest the best, precise and accurate websites or scientific literature to find the perfect novel druggable proteins or enzymes or even targets to initiate design and discovery of medicinal compounds?
See, again, the best things could be, but the best resource could be journals. So if you take a look at journals like Drug Discovery Today, now that's a review journal, right? As in, they don't publish research articles. So if you visit those, see, if you look at the issues, the latest issues, or say last 6 months or last 1 year issues, so they basically publish reviews of current interest, right? So when you take a look at a particular review, say the utility of FAD kinase inhibitors in cancer, right? So then they will talk about the FAD kinase as such, how that is an interesting target. They'll also summarize all the previous work done in that area, that includes the discovery of small molecules or macromolecules, or it could be phytochemicals, anything and everything. And then there will be, because it's a review article, it's a big reviews. Usually these reviews are very big. So then you go and refer to the cross references or the cited references, and then you will that will take you back to the original articles, where they have carried out all sorts of biological studies, or even for that matter chemistry, medicinal chemistry, structure activity relationship, maybe molecular docking, or other sort of computer aided molecular design. So my best suggestion would be, instead of relying on, as in, there are databases like there are platforms like Cortelis drug discovery intelligence platform. But then those are very, very expensive ones to subscribe, and probably academics, they don't really have access to that, or companies, even big pharma companies, probably not everyone would have them. So if you have access to those resources, where you get updated every day as to if a molecule moves from phase 1 to phase 2, or from preclinical to phase 1, or from biological testing to preclinical. So those are like, those databases are for industries, but then you can't really have access to them, because they are highly paid, or rather highly expensive, right? Paid databases. But then the literature, the latest literature, maybe even Journal of Medicinal Chemistry, the perspectives, if you look at the perspectives, they have got the latest information till date, right? Because not everybody can write review articles in for Journal of Medicinal Chemistry. It's only the experts who have worked in that area are only allowed to write review articles. They're not any Tom, Dick and Harry would write a review article. If you're not worked in that area, you shouldn't be writing the review article, because reviews are written by the experts only. I mean, if I work in an area for like last 10 years, I know the in and out of that particular area. So I am in a better condition to tell where the field is moving, where the field was, and what are the issues with the current technologies, and where how you can improve and all. So that's where the literature, like these good journals, that they are the best source, at least for students, for that matter. If you are in a company and if you have access to those databases, you essentially can definitely refer to those which will give you the best possible information. There are journals like drugs of future, drugs of today. You can also refer to that. So every article is like a monograph on a molecule which is probably in phase 3 or phase 2 or just approved drug. So very specialized information which contains everything about that drug. So those journals are also very effective. But I wouldn't suggest any free resource, or rather I don't know any free resource which will have all of this information. As in, you can search in Google and Google will give you some scattered information, but then the journals will have all the assimilated information at one page, and that's the best resource I would suggest.
Okay, we have one more question. Will adapting artificial intelligence help in speeding up the things associated with drug discovery and designing?
Definitely. In fact, it has already started making news. If you look around, you will see that for at least for COVID-19, there have been 2, 3 papers where they have claimed to discover, or rather come up with drug repurposing hypothesis with the help of artificial intelligence, or say DL and ML tools. So yes, they have already. In fact, if you read more about artificial intelligence, there are many specialized companies who are doing this day in and day out, as far as drug discovery is concerned. And it is just a matter of time. Let these 10 years pass, say in 2030, you probably will see that people will only talk of artificial intelligence, DL, ML for drug discovery, and all other techniques will probably take a backseat for sure.
Okay, so due to shortage of time, we are ending the question and answer session here. And now I would like to call upon Sainath to conclude this session with a vote of thanks. I think Sainath is facing some network issues, so
Okay.
uh, I'll present
Yeah.
the vote of thanks.
Hello. Yes, sir.
Okay, we have Sainath here.
Is it audible? Hello.
Yes,
Yeah.
yes.
Sorry for the inconvenience. Yes. Good evening, everyone. Good evening, sir. Good evening, everyone. Myself, Sainath from St. Paul's College of Pharmacy, Hyderabad. Thank you, Devika, for moderating this session. First, I'd like to thank our speaker, Dr. Prashant Karkar, sir, for accepting our invitation and sparing time from his tightly packed schedule. Thank you, sir, for such a wonderful session and enlightening all of with insightful presentation. Your words gives us more thoughts to learn more things about the drug discovery. Keep supporting us like this in all our future activities, and we would like to hear much more from you on different aspects of pharma related things in coming days, sir. And thank you again, sir, for answering all our queries with so much patience.
Yeah. Yeah, it's always a
We
pleasure
are
to.
really privileged to have you here today, sir.
Yeah, it's always a pleasure to interact with students, right? Yeah, so like I said, it's always a pleasure to interact with students, and I would be happy to be part of it, part of these activities, as and when you need me. If I am free, available, I would definitely would be happy to come and talk to you again. Yeah, thank you so much for the opportunity, and I do see a lot of familiar faces from NMIMS. Shreya is there, Varsha is there, of course, Devika is there. So yeah, it's good to see you guys, and thank you so much once again.
Thank you, sir. We would love to host you again very soon.
Yeah.
So I think we can end this meeting here. Thank
Okay.
you,
Thank you so
audience,
much.
for attending this session.
Thank you all. Take care and have a great weekend.